In this video, we will discuss about what are the reprogramming that has occurred within the pancreatic cancer cells because of the KRAS mediated mutations. So far, we have discussed that, or we have understood that most of the pancreatic cancer cases has this KRAS mediated mutations, or the KRAS mutations is more commonly found mutations in the pancreatic cancer patients. So how this KRAS mutations helps the pancreatic cancers towards acquiring its properties such as high proliferation rate and the aggressive metastasis. We will discuss that thing in this video, and we will cover some of the major points. So let us start with this macropinocytosis. So in this macropinocytosis, the proteins that are present in the extracellular matrix will be internalized within the tumor cells, and once the proteins are internalized they will be recycled through lysosomes, so that amino acids for which these proteins are composed of will be utilized towards maintaining the energy that is essential for this uncontrolled growth rate, and at the same time this metastasis and migrations properties. So apart from this macropinocytosis, there is another term that is autophagy. So macropinocytosis will recycles the proteins that are present in the extracellular matrix, whereas autophagy will recycles the proteins that are not necessary for maintaining these pancreatic cancer cells properties within the cells. So those proteins will be recycled through autophagy. So amino acids of this also will be utilized towards maintaining this pancreatic cancer cells properties. And apart from these two, there is another term which is mitophagy. So mitophagy is generally the recycle is means mitophagy means the recycle of the unhealthy mitochondria that are present within the cells. If they are not being taken care of, it will increase the ROS, that is reactive oxygen species, and increasing the ROS beyond a threshold level will make these tumor cells more sensitive towards the immune system. So in order to prevent themselves from the immune attack, or in order to evade the immune response of the host, the unhealthy mitochondria needs to be taken care of, and this will be taken care of through this increased mitophagy, and this will help in maintaining the low ROS within the tumor cells, and this will not trigger additional signaling pathways that will make the tumor cell sensitive towards the immune attack or immune detections. So once the cells reach the lysosomes, they will undergo autolysosomes where the amino acids will be recovered, and one of the important amino acids is glutamine that will help in increasing the nucleic acid and biosynthesis through mitochondria processing. At the same time, so this KRAS-related mutations uh, not only works towards these two properties, that is proliferation and metastasis. However, it will also work towards uh, preventing the tumor cells from being detected by the immune system. And one of the ways that it does this, it will release the proteins in the extracellular matrix or change the tumor microenvironment compositions in a way that the immune system will not be able to reach to the site of tumors. And one of the way that it does is by changing the pH in the extracellular matrix. At the same time, it will form a increased glycocalyx layer because of the reprogramming. And the glycocalyx layer that we have discussed many times in our previous videos is mainly composed of the carbohydrate structures, and because of the reprogramming that occurs in the tumor cells, the carbohydrate structures length will be high on the cell surface proteins that will cause or that will act as a physical barrier and prevents the immune cells to detect the non-self epitope that are expressed on the tumor cells. And since most of these carbohydrate structures, not all but most of the carbohydrate structures are resembles to the host carbohydrate structures pattern, cell surface proteins carbohydrate structures pattern, so immune cells will not be able to distinguish those carbohydrate structures from the between those carbohydrate structures between the normal pancreatic cells and the tumored pancreatic cells. So this will make the immune system difficult to identify and differentiate the pancreatic tumor cells and the normal pancreatic cells. So I hope you got some basic idea or brief overview about these KRAS mediated changes that occur in the pancreatic cancer cells. If you need to understand more about this reprogramming that occurs because of the KRAS mutations, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.